Hi, welcome to my 2-minute capsule on microbiology. So today I'll be discussing about Gram stain. So Gram stain was devised in 1884 by Hans Christian Gram. So what happens is the advantage of Gram stain is it helps to differentiate between two groups of bacteria. That means it differentiates bacteria into either Gram positive or Gram negative. It can be done within 2 minutes, so that from a sample, if we do a Gram stain, we know a provisional diagnosis whether it is Gram positive or Gram negative within 2 minutes. Based on that, we can start a treatment because the antibiotics for Gram positive and Gram negative are different. Now coming to the principle of Gram stain. So the Gram stain is done in 4 steps. So you have basically 4 different chemicals. The first one is carbofuction. So when you put it on a slide which contains a bacteria, the carbofuction enters into the cytoplasm and it stains the cytoplasm. Now what happens in a Gram positive bacteria, its cell wall is quite thick. Gram negative organisms, a bacteria cell wall is thin and it has a lot of lipids. So in the next step, you put a mordant, which has which makes the stain particles bigger. In the third step, you put a decolorizer, which is the most important step. So decolorizer shrinks the Gram positive bacteria and hence it closes down the pores. In Gram negative organisms, it has a lot of lipids. So the decolorizer actually dissolves the pores and opens up. So the initial stain it goes out, and when you put a counter stain, which is a different color, it goes inside the Gram negative bacteria, but cannot go inside the Gram positive bacteria. So Gram positive bacteria effectively retains the primary dye, Gram negative retains the secondary dye. So this is the principle of Gram staining. If you like my channel, please subscribe and tell me